Hello everyone, this is Mr. Schultz and this is AP Biology video 2.2. This video is going to cover the fluid mosaic model, which is the Biology in Focus chapter 5.1. So, the fluid mosaic model is talking about the cell membrane or the plasma membrane, and you're going to see both used equally because they end up meaning the same thing. The plasma membrane allows some substances to cross more easily than others, and we use the term selectively permeable. And what it means is it gets to pick what goes into and out of the cell. So selective meaning choosing, and permeability meaning allowing to pass through. The cool thing is the cell membrane is made up of a bunch of phospholipids, and these phospholipids are what are called amphipathic, which means that they have both a hydrophobic and hydrophilic region to them. So if you recall back to our first unit, when we talk about phospholipids, there's a head and two little tails. And the head likes water, it's hydrophilic, and the tails are hydrophobic. And when we put them into water, they automatically flip themselves so that the tails are all pointing inward and the heads are pointing outward. And they'll make a little shell in and of themselves. And due to the limited ability of water to pass through cells, a special protein is actually needed to allow water to pass through it. Water can pass through the cell membrane, but it needs some help a lot of times. It can fit through, but it's not fast enough to allow for certain nutrients and other things to get moved in and out of the cell, which is part of homeostasis. So the cell actually uses these proteins called aquaporins. And so here's a little picture of an aquaporin. And they allow for water to pass freely both into and out of the cell. Most membrane proteins are also going to be amphipathic, which means that they have both a hydrophobic and hydrophilic region, so that they can actually sit inside of the membrane itself. Because if they were one or the other, just hydrophobic or hydrophilic by themselves, they would either be pushed out or would never be able to get into or onto them. An example of when we look at a cell membrane, we talk about selective permeability, we can look at here. If it's a molecule that's too big or it's polar, it can't fit through very well. And so what happens is we need proteins to get them in. If it's small enough, like water, and even though it's polar, it can still kind of fit through, but it can't fit through very well. And so it needs help. And that's where we see things like proteins helping things move in and out. And then other things are just small enough or nonpolar, and they just pass through. Oxygen and carbon dioxide are examples of these. They're just small enough that they just kind of pass through the membrane without a problem. So, what do we mean by a fluid mosaic model? So, fluid means like a liquid. So, the membrane is a mosaic of protein molecules floating through a fluid bilayer of phospholipids. So, a mosaic is a bunch of small pieces that get put together to make a much bigger picture. So like if you look at this little sunflower picture at the bottom, that's a mosaic. It's a bunch of small tiles put together to make a bigger picture. The fluid mosaic model is the cell membrane is a bunch of small pieces put together to make a much bigger picture. And the proteins aren't just randomly distributed. They're actually clumped together in specialized areas for what that area of the cell membrane needs to do. You might be an area that moves salts in and out of the cell, or perhaps you're part of like a white blood cell that specifically works at engulfing a food particle or an invader. So when we look at the proteins, they're not just random. They're actually fairly well organized. How can a membrane, a bunch of small molecules, be fluid? Well, it's held mostly together by hydrophobic interactions. So when we look at the phospholipids, we see that they have a bunch of tails. And so these tails all sit together, and they're kind of what allow the interactions and the bonding to kind of stick together, and they allow them to kind of form like a liquid. And the phospholipids can really easily move lateral, which means that a phospholipid could go here, and it could just kind of crawl its way across, and they're allowed to kind of pass over each other or around each other. Proteins can move too, but they're much bigger and they tend to be embedded, and so they tend to be much slower when they move. And then the temp at which the membrane solidifies depends on its composition. When we look at the fluid mosaic model, we draw a whole bunch of little cholesterol molecules between the tails. We like to say that they kind of help keep the tails from locking up. And what happens is more unsaturated hydrocarbons mean a more fluid at lower temperatures. So if you're an organism, like an animal that lives in very cold temperatures, let's say a seal, you would expect to find many more unsaturated hydrocarbons in the cell membranes than you would all other organisms that say live in a tropical area. So you could actually look at like the health of foods in different areas, and you can see that animals that live in cold environments generally have more cholesterol in their food. Cholesterol also helps stabilize the effects of the temperature of the bilayer. So it's not just the hydrocarbons, but also the cholesterol itself. So just be aware that there's a couple of things that help stabilize and keep control of the actual membrane itself. When we talk about membranes, we also want to talk about where they come from. So the membrane lipid compositions appear to be adaptations to specific environmental conditions. An example would be that fishes that live in extreme cold will have more unsaturated tails. So if we look at the unsaturated tails and their bendiness, that kind of helps them from locking up, which means that the cells won't freeze as easily. Whereas archaea and bacteria that live in really hot conditions will often have nothing but saturated fatty acids, and they might also have special lipids that are built right into their membrane to help prevent them from like boiling alive, which is really interesting. Archaea are amazing. They can handle incredibly hot temperatures. Some organisms have membranes that change composition depending on the time of the year. So if you're an organism that lives in an area, like an environment or a climate that has very cold temperatures and very hot temperatures, like Wisconsin, you can see the composition sometimes changes over time. So be aware of that as well. On the AP test, you probably wouldn't be asked like, hey, what kind of phospholipids would this have? But you do need to be aware that the composition can change depending on the environment that an organism lives in. Now, the actual parts of the fluid mosaic that aren't phospholipids are things like proteins. So membrane proteins are going to be part of our mosaic. So the phospholipids are like a fabric, but the proteins actually determine the function of that part of the membrane, as well as the cell oftentimes. So cells that have specific jobs, like a nerve cell, will have very specific membrane proteins inside of them, like receptor proteins. There are two major membrane group types. There are the integral proteins. They penetrate the hydrophobic interior. So they actually go through the hydrophobic stuff. And then they can extend all the way through the membrane, or they might just be partially through it. So sometimes we see one that's kind of integral, but it isn't all the way through. It only goes halfway through. Peripheral proteins are not embedded. Instead, they're just kind of attached to the exterior. And so what that means is that they're kind of maybe hanging a little bit, or they might stick out a little bit. And they generally attach to the exoskeleton when they're inside of the cell, and then they tend to be on the ECM on the outside of the cell. So they're still attached to the cell, but they might not be entirely embedded into the membrane itself. Sometimes they stick in a little bit, and sometimes they're just completely just hanging loose. There are a number of functions for membrane proteins in and of themselves, and these can all be incredibly important. So here are just some of the functions. There's transport, moving things in and out of the cell. There's enzymatic activity. If you need to control a chemical reaction either inside or outside of the cell, you might find enzymes embedded into the actual cell itself. There can be proteins that can actually be attached to the cytoskeleton and the ECM, and they help hold the cell in place. They help hold organelles in place. Sometimes they help stabilize the membrane as well. There's cell-to-cell recognition, where cells can recognize other cells that belong or don't belong. So our immune system does this, where they look for very specific protein markers on the outsides of cells, especially glycoproteins. And if they have the wrong name tag on the cell, our immune system knows that they might be an invader and might start attacking them. There's also intercellular joining, where it could be it might allow for like water to pass through or a chemical of some kind for cells that are really close together and work together. And then there's signal transduction, which is how our cells can send signals either from one cell to another, or as they get a signal, passing that information down into the cell for transcription and translation to take place to make proteins. So it's just a response for the cells. So cell-to-cell recognition is one of the major functions of the cell membrane. It's the ability to distinguish one cell from another. It's very important in both embryo development and the immune system. And we might get into these a little bit more in later videos, but understand that cells need to be able to communicate and recognize each other. So cells recognize other cells through binding molecules. They could use what's called a glycolipid, which might be a like phospholipid with a little bit of a sugar added to it. In this case, glyco means sugar, so like a carbohydrate of some kind that's stuck to it, very specific usually, like a name tag, or glycoproteins, which is just a protein with a sugar molecule added to the top of it, which is also like a name tag. And depending on the system and the response you want, you might use glycolipids or glycoproteins. A good example of this is blood typing. So you have type AB, or O blood typing, and each one is determined by what markers you have on the outside of your blood cells. And so if you're type A and you get type B blood, your body recognizes that the little B markers on the outside of the blood are not where they should be. They don't belong. And so our immune system would attack them. So if you're type A blood and you get a B blood transfusion, this could kill you because your body will automatically attack these blood cells as they are an invader. And usually when you need blood, it's a life-threatening situation. And now you're putting blood in that's not good, and that could result in some really bad results. So you have to be aware of blood typing. Now, the nice thing is, if you have type O blood, you're a universal donor, which means that I'm type O blood, which means I can donate my blood to anybody who's positive, and that's our B factor, and we're not going to worry about that. But basically, I'm almost a universal donor. Most people can take my blood as long as they have a positive type blood. AB is the weird one. AB can take any kind of blood. They're a universal acceptor, which is really cool. So this is the end of the plasma membrane notes. It's a nice quick video. Hopefully it's helpful for you. If you have any questions, by all means, ask in class, and I'll be more than happy to talk about the cell membrane and its functions.